Hello, my name is Jason Adrians. Today is 5/8/2025, and this is my week 13 reproductive system portfolio. I decided to do the case 2 study on male infertility following testicular injury. So this is a presentation. I'll be walking through the presentation and information about this topic throughout the video. So as I mentioned, the entire purpose of this presentation is to figure out why male infertility happens after testicular injury. And in order to answer the three questions posed, being how do the testes become externalized during development, why might lymphocytes invade the germinal epithelium following injury, and why might this condition result in sterility, you kind of need to understand how sperm gets generated in the first place. So that's what spermatogenesis, or that's the purpose of this slide, is to demonstrate what spermatogenesis is and to provide an overview of it. So spermatogenesis happens within the seminiferous tubule. If you follow my mouse here, the seminiferous tubule is within the testes, and it starts with a germ cell. So if you follow this diagram here, this germ cell goes through mitosis and it splits, and it has the exact same number of chromosomes that it did in the first place. So it's identical cells. So it's 2N to 2N. And then it splits and it has a type A spermatogonium and a type B spermatogonium. And the type A one actually just goes back up and will eventually split again. But the idea behind this is so that it never runs out of the germ cell or the stem cell that ultimately goes through spermatogenesis. And the type B cell actually goes on and goes through spermatogenesis. So it goes down and it becomes a primary spermatocyte. This primary spermatocyte goes through meiosis 1. This diploid cell, actually, this is where it becomes a haploid cell. So then that becomes a secondary spermatocyte. And then the secondary spermatocyte goes through meiosis 2, and then it ends up with 4 haploid cells. And these 4 haploid cells are called spermatids. So this whole process is actually called early spermatogenesis. And then late spermatogenesis is where these 4 haploid cells actually start becoming sperm cells or spermatozoa. It's a very complex process through this all, but that's kind of an overview of it. And the key point that I want to make here is that it does happen within the seminiferous tubule, but the seminiferous tubule is made up of Sertoli cells. This whole process doesn't actually happen within the Sertoli cells. It happens intracellularly, meaning in between the cells, and the Sertoli cells kind of regulate and provide nutrients for this process to happen. And also a key point is that why the testes are located outside of the body is because it requires a lower temperature, and this whole process here wouldn't be successful, or the spermatozoa would actually die if it was in the normal body temperature of 37 degrees Celsius, and so it requires a slightly lower temperature at roughly 34 degrees Celsius to actually happen and to maintain the spermatozoa so they don't die ultimately. Something else that I forgot to mention that's very important about this is, as I mentioned, this process happens intercellularly, so it's in between the Sertoli cells. But the Sertoli cells actually provide a barrier in between the blood supply to the seminiferous tubule and the lumen. So the side of the seminiferous tubule that's closest to the connective tissue, or the basal side, is where this process happens, where the germ cell goes through mitosis and then ends up with a type A spermatogonium and a type B spermatogonium. Then there's the adluminal side, which is closest to the lumen, and that's where this process happens, where the primary spermatocyte ultimately gets down to the spermatozoa, and then it's released into the lumen. But there's a barrier that's created, and I'll get into more why that's important later on. But there's a barrier created called the blood-testes barrier, and that's a very important thing. So I'll be talking about that more later on, but I just wanted to mention that here just so it gives an overview of it. So I intentionally put the hormonal feedback loop after this spermatogenesis process because this hormonal feedback loop is ultimately what starts and stops the spermatogenesis process. So it all starts in the brain with the hypothalamus. The hypothalamus secretes gonadotropin-releasing hormone onto the anterior pituitary, or GnRH for short. GnRH then goes to the anterior pituitary, and the anterior pituitary releases two hormones, luteinizing hormone and follicle-stimulating hormone. Luteinizing hormone goes to the testes and acts on the Leydig cells or the interstitial cells. And then the interstitial cells produce testosterone. And testosterone does two things. It stimulates sperm production, but it also inhibits the process of, or inhibits production of GnRH and LH. I'll get into that more, but I'll focus on sperm production for right now. So testosterone stimulates sperm production. Going back to the anterior pituitary, the hormone FSH that it releases goes to the testes. And FSH stimulates the Sertoli cells to start secreting things into the intercellular space to start spermatogenesis, as well as testosterone. And then spermatogenesis starts to happen, and sperm starts to go into the lumen of the seminiferous tubules. The Sertoli cells have things on them to figure out when sperm count is high. And they have receptors on them to figure out when sperm count is high. And when sperm count is high, the Sertoli cells start to secrete a protein called inhibin into the bloodstream. Inhibin then goes to the anterior pituitary and to the hypothalamus to inhibit the production of GnRH and FSH. And ultimately, if you inhibit the production of GnRH, then you will inhibit the production of FSH. And testosterone, as I mentioned before, it stimulates sperm production, but it also inhibits the production of GnRH and LH. So yet again, if you inhibit the process or inhibit the production of GnRH, then you're going to inhibit the LH release from the anterior pituitary. So it's a negative feedback loop that happens there from testosterone. I think that's the basis of the hormonal feedback loop. So after briefly running through those two processes, the first question you might have in the overview of the whole presentation of how testicular injury can cause infertility is, why are the testes residing in a place where they're so vulnerable to injury? So how do the testes become externalized? The testes are externalized because the sperm, that's where sperm production happens, and sperm are very sensitive to temperature. So at normal human body temperature of 37 degrees Celsius, sperm actually die. So in order to control the temperature or to lower the temperature better, they reside outside the body. And that temperature is trying to stay around 34 degrees Celsius, which is 3 degrees cooler than normal human body temperature. So while they reside in a more vulnerable place, that's the reason they need to. So during gestation, the testes start near the kidneys inside the belly. And then the gubernaculum pulls them down into the scrotum before birth. And then the gubernaculum actually really doesn't have a purpose after this, but it still does reside within the scrotum. You can see it in adulthood, but it doesn't really have a purpose other than just to pull the testes down into the scrotum before birth. And then the scrotum, as I mentioned, helps keep them cooler, which helps sperm development. So the next question is, why do lymphocytes invade after injury? So you got to remember that the whole purpose of this presentation is yet again to figure out why testicular injury can cause infertility. So going all the way back to the spermatogenesis overview, the spermatogenesis happens intercellularly, in between the Sertoli cells. And the Sertoli cells have special things to separate the spermatogenesis process. So there's the mitosis process, and then there's the meiosis process, and all the way through to the spermatozoa. And as I mentioned in that, I briefly mentioned that there's the barrier, the blood-testes barrier. That barrier is very important because spermatozoa don't actually start getting created until puberty. So the adaptive immune system that humans have don't recognize sperm because they weren't there from birth. The germ cells were, but the sperm itself was not there. So if sperm got into the bloodstream, the body would recognize that as foreign and start attacking it, which is not good. So this blood-testes barrier keeps the blood and the sperm separate. so that the body does not recognize sperm because it's never in the blood. However, after injury, the barrier can be broken, and then blood can get into the testes, or the sperm can get into the blood, and then the body will see sperm as foreign and attack them, which then lowers sperm counts massively, which is ultimately what creates infertility or sterility. So as I mentioned, that barrier is what really keeps everything separate, and if that barrier is broken by a blunt trauma or something like that, then that's what ultimately causes the sterility or infertility, because the immune response to sperm will be to destroy it, which obviously you don't want, but that's just the body's natural way of thinking that it's helping. It's not, but that layer has been broken, so it's recognizing sperm as foreign, so it's going to destroy it. Thanks for listening.